Hi, my name is Jennifer Waters, and I'm a graduate student in the House Lab at San Diego State University. Thank you for coming to my poster on the role of IGFBP5 in obesity-related ovarian cancer progression. So before we get into the data, I wanted to give a brief background. Ovarian cancer is the most lethal gynecological malignancy with increased risk and worse overall survival among patients with obesity. And this is interesting because ovarian cancer metastasizes preferentially towards the omentum, a fatty organ that is composed primarily of fat cells or adipocytes. However, both in obesity and in cancer, the adipocyte precursor cells, also known as preadipocytes, become more prevalent. And it's not known what effect this increase in preadipocytes has on the tumor microenvironment. So we sought to understand how preadipocytes influence ovarian cancer cell behavior. We first performed some preliminary in vitro co-cultures with cancer cells and primary omental preadipocytes derived from obese donors and measured the effect of preadipocytes on colony formation, which is an assay for cancer cell survival. And as shown in panel A, we saw that preadipocytes attenuated an increase in colony formation. So with this in mind, we then moved into a subcutaneous xenograft model in mice and observed that preadipocytes increase the tumorigenic potential of ovarian cancer cells in vivo, specifically by increasing the tumor-forming potential of a low dosage of cancer cells in conjunction with a high dosage of preadipocytes, which is denoted by the 1:40 in the bottom of panel B, indicating the ratio of cancer cells to preadipocytes that were injected in each condition. And this tells us that low dilutions of cancer cells are more tumorigenic in the presence of an abundance of preadipocytes, which is representative of the obese tumor microenvironment. This is expanded upon in panel C, where we used a transplantation assay that used tumor cells isolated via fluorescence activated cell sorting from the 1:40 tumors and re-injected them again, with and without preadipocytes, and saw that sustained signaling from the preadipocytes is required to maintain this phenotype. To explore the mechanism by which preadipocytes support tumor initiation, we performed RNA sequencing in co-cultures that recapitulate our mouse model. And we observed an increase in ECM remodeling genes marked in orange. But notably, the most significant differentially expressed gene was insulin-like growth factor binding protein 5, or IGFBP5, which is circled in red. When we explored the clinical significance of IGFBP5 in the Cancer Genome Atlas, we discovered that increased expression of IGFBP5 in patients is significantly correlated with decreased overall survival. Studies are currently underway to further investigate the role of preadipocyte-induced IGFBP5 in supporting ovarian cancer progression. And with that, I would like to thank the HealthLINK Center and my other funding sources. Thank you very much.